Dear viewers, Assalamu alaikum. May Allah shower you all with His blessings. I welcome you all to my channel AmreenaMehrajTutorials. I dedicate my channel to all those learners who wish to clarify their concepts regarding fundamentals of biology. If Allah wills, I'll be uploading a series of lectures pertaining to class 11th and 12th biology. These lectures will be helpful for you to qualify board level examinations, be it CBSE or any board for that matter. Even these lectures will help you to qualify medical entrance examinations like NEET and AIIMS or any medical entrance examination for that matter. Do like, share and subscribe my channel so that I can reach to more and more learners, inshallah. Let's get started. Today we'll begin with class 11th and we will start with zoology portion. Out of zoology I have selected unit 2nd, that is cell structure and function. My dear students, when we talk about this unit, it has got different components. In this unit, you are supposed to study about cell and its organelles. Then you are supposed to study about biomolecules. Afterwards, you are supposed to study about cell division, how cell divides, both by means of mitosis as well as meiosis. Then you are supposed to study about enzymes, which are biological catalysts. So in this unit, we are supposed to study about all these things. Now, the best thing to begin this unit is to have this information about biomolecules. It's very important to understand biomolecules first. Then we will skip to cell and its organelles. Then we will study about cellular division, and at last we'll study about enzymes. So today we are going to begin with biomolecules. So my dear students, you have to remember that this unit we are going to cover in different stages. Stage number 1 is biomolecules. Let us understand about these biomolecules in detail. To make it very simple for you people, it's very easy for you to get an idea about what biomolecules are. All the molecules which are present, so I'll encircle over here, all the molecules which you will find in a living system. Bio indicates what? A living system. All the molecules which you will find in a living system. What is a living system actually? It's a cell. So all the molecules which you will find inside a cell, they are biomolecules. What will you find inside a cell, my dear students? From your basic knowledge, you know that a cell has got what? Basic thing which you will find in cell is water molecule. Then you will find various ions like sodium, potassium, calcium, chlorine. You can call all these substances what? You can call them biomolecules because you find them in a cell. Similarly, thinking in a higher level, you can recollect your information about these. Carbohydrates. You might be knowing about lipids. You might have an idea about proteins. You might have an idea about DNA or RNA. What are these? They are also biomolecules because you find them all wholly and solely in which? You find them in a cell. So what are biomolecules? To put in a simple statement, all the molecules, be it water, be it ions, be it complex substances like carbohydrates, lipids, proteins, DNA, RNA, all of these molecules are called biomolecules. Isn't it very simple? Now, moving ahead. How do we came to know about these biomolecules and how can we categorize them? That's also very interesting. Let us, for that matter, will perform a chemical analysis. It's very interesting. Let us, my dear students, let us take a sample. Which sample you are supposed to take? You can take a animal tissue. Say for example, you can take a piece of liver or you can take a potato. an animal tissue or a plant tissue will work for this analysis. Now what you are going to get out of this analysis, you will come and understand. So take this sample, let us take this liver sample. Then you are supposed to add a solvent to this sample. The solvent which we are using over here is trichloroacetic acid. So we are using this solvent and we are adding it into the sample. Then we will grind this tissue. So you are using this solvent and in this you are placing this sample, that is the liver tissue, and then you are grinding it. In a simple mortar and a pestle you grind it. What you will get? You will get a thick slurry, you will get a paste kind of a thing. Okay? So you will get this grinded paste of liver in this solution. What is that solution made up of? Trichloroacetic acid. Now you are supposed to take this slurry and you will perform a simple task of filtration. What you do over here, you take a simple cheesecloth or you can take cotton. Then you are supposed to take this slurry and you are supposed to place it on that cloth. And what will happen simply? Filtration will start taking place. Substances which will be very bigger, they will remain on the cheesecloth and a solution will pass down and you will get a filtrate. So after filtration you will get two components. Number one, you will get filtrate. It will be the solution which will come down and some of the part of the slurry which will remain on the cheesecloth. That part is called, what is that called? It's called retinate. So it will be the solid mass that will remain on the cheesecloth. Now, can you get this idea what filtrate is? It's all those molecules of the liver which were soluble in trichloroacetic acid and they got filtered in this filtrate. Can't we call this filtrate then acid soluble pool? So all those substances, all those molecules which were present in the liver tissue, they got grinded and they got filtered along with trichloroacetic acid and we obtained them in an acid soluble pool. Whereas all the substances which do not get solubilized in trichloroacetic acid, they remain in the retinate. So we can call them what? Acid insoluble. You can call that acid insoluble pool. So all these molecules which are soluble in acid, you will get them in this filtrate part, and all the substances which are not soluble in acid, they will remain in retinate part. There is one more thing you can guess over here. These molecules which are present in filtrate, they got filtered because their molecular weight was less. How much less it was? It was less than 1000 Dalton. So all the molecules which are present in acid soluble pool, their molecular weight was less than 1000 Daltons. Whereas all the molecules which were in the retinate, that is the acid insoluble pool, their molecular weight was more. How much? It was more than 1000 Dalton. So this is the idea you need to get. Scientists call these molecules which remain in the filtrate part, scientists call them biomolecules. And all the molecules which remain there, they are also called what? Biomolecules. But there's an interesting thing. Molecular weight of these biomolecules which remain in the filtrate, molecular weight is less. And over here, the molecular weight of these biomolecules which remain in the retinate, the molecular weight is more. Here it's less than 1000 Dalton, there it's more than 1000 Dalton. Accordingly, these molecules were called microbiomolecules and these were called macrobiomolecules. Macrobiomolecules. So you have two terms. All the biomolecules with less than 1000 Dalton molecular weight, they are microbiomolecules. All the molecules whose molecular weight is more than 1000 Dalton, they are called macrobiomolecules. So we got two types of biomolecules, microbiomolecules and macrobiomolecules. Now, in order to understand more, we need to think like this. Microbiomolecules are comparatively what? They're very simple because you got them in which portion? You got them in filtrate part. They're simple, whereas these are comparatively complex as far as their conformation is concerned, as far as their design is concerned. These are simple and those are complex. Now, looking at their molecular weight is less than 1000 Dalton. To be more specific, the range is 18 to 800 Daltons. So these molecules range in their molecular weight from 18 to 800 Dalton, whereas these have molecular weight more than 1000 Daltons, comparatively bigger. If you will find these in this category, you will find biomolecules to be both organic as well as inorganic. So my dear students, microbiomolecules, nature-wise, they could be organic as well as inorganic. But macrobiomolecules are specifically organic in nature. They are organic in nature. Moving ahead. Now, what is the type of biomolecule which falls over here? They're very simple. My dear students, simple carbohydrates. Simple carbohydrates, which you also call sugars. They come under microbiomolecules. Do you know about glucose, fructose, galactose, sucrose? They're all simple sugars. They all fall under microbiomolecules. You call them simple carbohydrates. Whereas, if these simple carbohydrates or simple sugars are bound to each other and if they form long chains, then you call them what? Complex carbohydrates. These are not then micro, they are macrobiomolecules. Very simple. Similarly, you might know that in your cell what are present are amino acids. Amino acids are microbiomolecules. Whereas, if you will join different amino acids, they'll form long chains, long polymers, which are called proteins. They fall under macrobiomolecules. Similarly, you might know about these. You might know about nucleotides. Like amino acids are the building blocks for proteins, nucleotides are the building blocks for your DNA and RNA. So nucleotides fall under microbiomolecules. When you link them together, when you chain them together, you'll get long chains of nucleic acids. That is DNA and RNA. These nucleic acids then form the macrobiomolecules. There is one specific thing. They are lipids. Lipids technically are microbiomolecules because their molecular weight is less than 800 Daltons. But while we were performing that grinding of liver tissue, all the lipids, they were converted in the form of a vesicle. Those vesicles do not undergo filtration. They remain in the retinate. Do you remember retinate? Retinate was that part which remained upon the cheesecloth. So, lipids do not get filtered. They do not come down along with the acid. They remain within the retinet because they are not, broken down into individual components. They remain in the form of big vesicles. So on that note, lipids are placed under macrobiomolecules. They are placed under macrobiomolecules, but technically these are actually microbiomolecules. So why do we put them here? Because you do not get individual lipids in the filtrate. You will get lipids in the form of big vesicles which remain in the retinet part. That's why during filtration process you will not get these lipids in this particular fraction. They remain a part of the fraction which includes macrobiomolecules. So you need to remember this thing, that lipids technically they are micro. Their molecular weight is less than 10,000 Dalton. According to this, they are what? They are micro, but they do not come under this pool. What was this pool? This was acid soluble pool. They do not come under this pool. They do not get filtered in this pool. They remain in the other pool, which is what? Which is acid insoluble pool. This is a very important point. So you need to get this thing clear, that lipids, although having less molecular weight, they do not come to the acid soluble pool. They do not get filtered in this acid soluble pool because they are in the form of big vesicles and they remain as a part of acid insoluble pool. So what do you get to understand from this? That you have variety of biomolecules. You have simple carbohydrates, you have amino acids, nucleotides, you have complex carbohydrates, proteins and nucleic acids. We are supposed to discuss about each biomolecule in detail. Dear students, other than organic and inorganic these substances, we need to analyze that cells also contain basic elements. Okay? We can come to understand about those elements by performing ASH analysis. So if you want to get this information about what? Elements which are present in the cell, you can perform ASH analysis. This is also very simple. Here you will again take a tissue. You are supposed to let this tissue dry completely so that you will get its dry weight. How you will get the dry weight? You will let it dry so that all the water will evaporate. Then you are supposed to burn this tissue. You can burn it in oven, so you have to give it dry heat. You have to burn it in an oven. Once you burn it, what will happen? All these gaseous or what you call volatile organic substances, they'll be what? They'll be removed completely. And you'll be left with ash. This ash is going to contain all inorganic oxidized elements. This ash is analyzed and it was found to contain different elements. And scientists have got to know that elements are present in our cells in different percentages. Some are present in more amounts, so we call them macro elements, which include carbon, hydrogen, oxygen, nitrogen, sulfur and phosphorus. So these are the elements which are present in more percentage in our cells. Similarly, some are present in less amount. We call them micro elements. These can include elements like sodium, potassium, calcium, chlorine, iron, etc. And there are certain elements which were present in the cell, but the quantity was negligible, or you can say they are present in trace amounts. So you call them trace elements. Trace elements include silicon, cobalt, molybdenum, manganese, etc. So till now you got this idea that cells contain microbiomolecules like simple carbohydrates, amino acids, nucleotides, etc. Macrobiomolecules like complex carbohydrates, proteins, nucleic acids, etc. Similarly, cells have different elements in different proportions about whom we have come to know through ASH analysis. Let's sum up what we have learned in lecture number 1. Out of lecture number 1, we have learned about chemical analysis, we have learned about ASH analysis, then we have come to understand about microbiomolecules, we have learned about macrobiomolecules. In our next video lecture, inshallah, we'll begin with the first biomolecule. They are carbohydrates. We will discuss about their physical properties, their chemical properties, and we will classify them in different categories. You will all come to know that in our next video. So stay tuned. Till then, take care of yourselves and don't forget to like, share and subscribe my channel. Take care. Fi amanillah.